In this presentation, we are going to learn about tumor immunology. We have seen these topics. In this presentation, we are going to learn about innate lymphoid cells and the effects of aging and stress on immune system. Tumor immunology. On their surfaces, tumor cells can reveal antigens that stimulate the immune system to destroy the tumor. When cancers develop, this immunological surveillance system, primarily the function of T cells and natural killer cells, has failed to prevent the growth and metastasis of the tumor. The topics again: innate lymphoid cells first, and then the effects of aging and stress. After watching this presentation, you will be able to explain the relationship between the immune system and cancer. Oncology means the study of tumors. Oncology has revealed that tumor biology is similar to and interrelated with the functions of the immune system. Most tumors contain clones. What clone is? Clone is a population of genetically identical cells. So, the clones formed from single cells that divide in an unregulated fashion, in contrast to normal lymphocyte clones that are subject to complex inhibitory controls. However, newer techniques have shown that primary tumors eventually become composed of subpopulations that can be distinguished by their different mutations and that may differ in their responses to cancer therapies. When cells become cancerous and divide uncontrollably, they de-differentiate. It means that they lose their specializations to become more like the cells of an embryo. Tumors are described as benign when they are relatively slow growing and limited to a specific location. For example, warts. Malignant tumors grow more rapidly and undergo metastasis. Metastasis is a term that refers to the dispersion of tumor cells and the resultant seeding of new tumors in different locations. The term cancer, as it is generally applied, refers to malignant tumors. The word cancer, in Latin crab, stems from Hippocrates' observation of a tumor being fed by blood vessels which resembled the claws of a crab. Cancer is now believed to result from altered expression of oncogenes, the genes that promote cancer, tumor suppressor genes, and genes that code for microRNA. As tumor cells differentiate, they reveal surface antigens that can stimulate the immune system to destroy the tumor. Consistent with the concept of differentiation, some of these antigens are proteins produced in embryonic or fetal life and not normally produced postnatally. Because they are absent at the time immunological competence is established, they are treated as foreign and fit subjects for immunological attack when they are produced by cancerous cells. The release of two such antigens into the blood has provided the basis for laboratory diagnosis of some cancers. Carcinoembryonic antigen tests are useful in the diagnosis of colon cancer, for example, and tests for alpha-fetoprotein, normally produced only by fetal liver, help in the diagnosis of liver cancer. The immune system can guard against cancer by recognizing antigens that are expressed in malignantly transformed cells. For example, carcinogens cause mutations, and these mutated genes can produce tumor-specific antigens. Also, some cancers are caused by viruses and so can display viral antigens that flag tumor cells. Examples include antigens of the human papilloma virus, which can cause cervical cancer, and antigens of the Epstein-Barr virus, which can cause Burkitt's lymphoma and nasopharyngeal carcinoma. Even normal self-antigens, if they are overexpressed due to gene duplication, may trigger attack by the adaptive immune system. In cases where tumor cells do not display antigens that directly stimulate T and B lymphocytes, the tumor cells may still activate the natural killer cells of the innate immune system. The adaptive immune response could then be triggered by molecules generated during the inflammation caused by the innate immune response to the tumor cells. Dendritic cells can present tumor antigens to cytotoxic T lymphocytes at the site of the tumor, or they can migrate through lymphatic vessels to cytotoxic T lymphocytes in lymph nodes, activating the T cells to kill the cancer cells that display the antigens. The concept of immunological surveillance against cancer was introduced in the early 1970s to describe the proposed role of the immune system in fighting cancer. According to this concept, tumor cells frequently appear in the body, but are normally recognized and destroyed by the immune system before they can cause cancer. So, in this figure, you see that mm, cytotoxic T cell induces apoptosis in a cancer cell. So apoptosis of the cancer cell is shown by its budding vesicles or apoptotic bodies. Innate lymphoid cells. Innate lymphoid cells are a more recently discovered class of cells that are part of the innate immune system, even though they are microscopically indistinguishable from the more numerous lymphocytes of the adaptive immune system. Unlike the lymphocytes of the adaptive immune system, innate lymphoid cells do not have receptors for specific antigens on their surface and do not respond in a specific way to antigens. Instead, they respond quickly to signals from infected or injured tissues as part of the innate immune system. There are two subclasses of innate lymphoid cells: non-cytotoxic helper cells and natural killer cells. Non-cytotoxic innate helper lymphoid cells. First, these cells are the innate immune system's equivalent to the T helper T cells of the adaptive immune system. In response to infections and tissue damage, they secrete various cytokines that promote an immune response. They are prominent in the intestine and other mucosal barriers exposed to commensal as well as potentially pathogenic microorganisms, where they help shape a proper immune response to such pathogens as viruses, bacteria, and fungi. Now the second, natural killer cells. Natural killer cells are analog to the cytotoxic T cells of the adaptive immune system, but they lack the receptor diversity generated by gene rearrangements to provide receptors for specific antigens. Instead, they inherit receptors that allow them to target malignantly transformed cells, meaning cancers, and cells infected with intracellular pathogens such as viruses. They also have receptors that interact with class 1 major histocompatibility complex molecules on the person's own cells, giving them tolerance to the person's self-antigens and preventing autoimmune attack. The natural killer cells were discovered by their ability to restrain the production of tumors in a strain of mice that genetically lack a thymus gland and T lymphocytes. Resting natural killer cells release cytokines including interferon gamma and others. They also contain intracellular granules with coenzymes and perforin. Thus, like cytotoxic T lymphocytes, they can destroy target cells by cell-to-cell contact. Unlike cytotoxic T lymphocytes, natural killer cells can do this efficiently without prior exposure to the foreign antigens. However, in order for natural killer cells to be fully effective, they must first be activated by interferon alpha, interferon beta, and other pro-inflammatory cytokines. Once activated, the natural killer cells release interferon gamma and other cytokines that help activate macrophages and the cells of the adaptive immune system. Because natural killer cells do not require prior exposure to foreign antigens, they can provide a first line of innate cell-mediated defense that is subsequently backed up by the adaptive specific immune responses of cytotoxic T lymphocytes. Interestingly, recent research has shown that natural killer cells, like the cells of the adaptive immune system, can produce long-lasting memory cells and mount a stronger secondary response to a subsequent challenge. Thus, although natural killer cells are considered part of the innate immune system, they can have characteristics of the adaptive immune response. The discovery of a type of T-cell scientists have dubbed natural killer cells, which respond to lipid changes and have both innate and adaptive immune characteristics, also blurs the distinction between these divisions of the immune system. Now, effects of aging and stress. Susceptibility to cancer varies greatly. The Epstein-Barr virus that causes Burkitt's lymphoma in some individuals, mainly in Africa, for example, can also be found in healthy people throughout the world. Most often, the virus is harmless. In some cases, it causes mononucleosis, meaning involving a limited proliferation of white blood cells. Only rarely does this virus cause the uncontrolled proliferation of leukocytes characteristic of Burkitt's lymphoma. The reasons for these differences in response to the Epstein-Barr virus, and indeed for the differing susceptibilities of people to other forms of cancer, are not well understood. It is known that cancer risk increases with age. According to one theory, this is because aging lymphocytes gradually accumulate genetic errors that decrease their effectiveness. The functions of the thymus also decline with age in parallel with a decrease in cell-mediated immune competence. Both of these changes, and perhaps others not yet discovered, could increase susceptibility to cancer. Numerous experiments have demonstrated that tumors grow faster in experimental animals subjected to stress than they do in unstressed control animals. This is attributed to the stress-induced increase in corticosteroid secretion, which suppresses the immune system. Indeed, the immunosuppressive effects of cortisone are used medically for treating chronic inflammatory diseases and for reducing the immune rejection of transplanted organs. Some recent experiments, however, suggest that the stress-induced suppression of the immune system may also be due to other factors that do not involve the adrenal cortex. Future advances in cancer therapy may incorporate methods of strengthening the immune system into protocols aimed at directly destroying tumors. Now, checkpoints. Explain why cancer cells are believed to be dedifferentiated and describe some of the clinical applications of this concept. So, cancer cells are dedifferentiated because cancer is believed to result from altered expression of oncogenes, tumor suppressor genes, and genes that code for microRNA. As tumor cells they differentiate, they reveal surface antigens that can stimulate the immune system to destroy them. So consistent with the concept of the differentiation, some of these antigens are proteins produced in embryonic or fetal life and not normally produced in postnatal period because they are absent at the time of immunological competence is established, meaning immunological tolerance is developed to the proteins already present in the body. So, since these proteins are not present at that time, they are accepted as foreign proteins, and they are subject for immunological attack. So, the release of these types of antigens to the blood provides the basis for laboratory diagnosis of some cancers. Here are the two examples. First, carcinoembryonic antigen test is positive in colon cancer, and alpha fetoprotein antigen is present in liver cancer. Define the term immunological surveillance and identify the cells involved in this function. So immunological surveillance is following the cancer cells. When cancer cells appear, they are recognized and removed by the immune system cells before causing cancer. So this is called immunological surveillance, proposed in 1970s. The cells taking part in this immunological surveillance are non-cytotoxic helper cells and natural killer cells. Non-cytotoxic helper cells behave like helper T cells in adaptive immune system, whereas natural killer cells they have receptors that make them able to recognize any transformed cancerous cells and destroy them. These natural killer cells, however, they do not attack our own healthy body cells because our own body cells, healthy cells, they have major histocompatibility complex 1 proteins on them. Explain the possible relationship between stress and susceptibility to cancer. So the studies have shown that cancer tissue in stressed animals are growing faster compared with unstressed animals. It may be because of excessive release of cortisol from cerebral cortex in stressed animals, and cortisol suppresses the immune system so that cancer cells can grow faster. So now we have finished our subject, tumor immunology. Thank you very much for your attention.